Okay, good morning to one and all. Today we'll start with the next practical class that is about the introduction of biochemical test. So introduction of biochemical test, in this class we will move like this, that first I will tell you about the importance of biochemical test, particularly in microbiology. Then I will give a glimpse about the basic chemistry, that how these biochemical tests are performed, or what is the basic theory behind performing this biochemical test. And at the end, we will try to we'll try to understand the principle, procedure, observation and interpretation of this four biochemical test. Other biochemical test I will take up in next part, that is biochemical test part 2. Okay, so let me start with the first, that is importance of biochemical test. So there are three important points. The first and most important is identification. So earlier class we had taught that how to collect the sample, how to send the laboratory, then how we can isolate the bacteria. There might be a non-fastidious or fastidious. There might be selective, non-selective media. So depend on many factors, we have to choose the appropriate media, appropriate sample, we have to process, and we have to find out that this particular bacteria are actual the pathogenic one, and these are the actual responsible for causing disease in particular target host. Now once we are identifying, then your jobs is not completed. You have to give that which genus, which species, whether it is a new genus or new species. Then you have to classify. You may require to differentiate from closely related organism. So a battery of test is required beside the isolation or beside the morphological characteristic of colony morphology and bacterial morphology. You require this battery of biochemical test to find out or to classify this unknown bacteria. So generally what we are doing, after collection of sample, we are going for isolation. After isolation, we are going for pure culture. Pure culture, sir might have covered in depth. So pure culture is also important technique. Once we are getting a pure culture, then we can go for colony morphology. We get some idea based on that colony morphology. Then we go for bacterial morphology. So bacterial morphology will add up something to colony morphology, and then we have to perform battery of biochemical test to reach final identification, differentiation and classification. For example, some unique enzymes are present in some specific genus. So if we are demonstrating that these enzymes are present in particular isolate, then we can classify or identify them as a specific genus. Similarly, if you are performing some test, if that is present for one group of bacteria and that is absent for closely related other group of bacteria, then that will help us in differentiation of these two bacteria. Because if you are seeing gram-positive cocci, then gram-positive cocci might be a staphylococcus or streptococcus. So you need to differentiate these two based on biochemical test that we will see in coming slide. And last, that is a classification. So it also helps or add up in the classification of unknown bacteria or the isolate that we have isolated from the field sample. So this is the importance of biochemical test in the microbiological study. And nowadays molecular tests are replacing some of biochemical tests because of some because of some heritable lacunae, or it has some disadvantage. So over the biochemical test, there are many molecular tests nowadays relied for classification and identification, differentiation. But still we cannot absolute, or we cannot remove the biochemical test from the microbiological study. So
Hello, sir.
with this we will move to the next, that is about the basic chemistry of the test. So how this, everyone learned about the substrate enzyme reaction. So what happens that enzymes are specific to some genus of bacteria. So enzymes are specific to some genus of bacteria, and presence of enzyme is demonstrated by some substrate. So what we are doing to demonstrate the presence of this enzyme in specific bacteria, we are providing some substrate. If this substrate is specific to enzyme, that will bind, and ultimately reaction will happen, and that will lead to product formation. So this product formation might be anything, and this product formation ultimately visualize using some indicator. So if indicator indicate that this product is formed by after addition of this substrate, then indirectly we can say that particular enzyme is present in this sample. So if enzyme is present, we can say that this particular bacteria or this enzyme is indicative of this genus of bacteria, and we can say that the isolate might be of this particular genus. So basic chemistry is based on substrate product or substrate enzyme reaction. So now move to the next, that is what type of enzymes are present. So generally they are broadly classified into one that is based on site of action, and second according to enzyme production. So endo and exo, these are two. Endo means inside, they are present inside the cell wall, and exo means they can secrete or they are present outside the cell. They are known as exo enzymes. So there are the tests they are targeting endo enzymes, and there are some tests they are targeting exo enzymes. The second, that is enzyme production. So sometimes they are inducible. Inducible means we require some external signal to induce the production of that enzyme before performing that test. And in contrast to this, there are some enzymes which are constitutive. Constitutive means they are constantly produced. They do not require induction before performing test. So these are the basic types of enzyme we are targeting in biochemical test. Now, these are generally kind of bacterial enzymatic reactions. Sometimes we are performing these tests on some toxic waste. Sometimes we are performing on reduction or oxidation of particular group. Sometimes we are also targeting some amino acid and their degradation. And ultimately we are seeing whether this particular amino acid degrade or not. And some biochemical tests are present on type of utilization of carbohydrate. Because bacteria, they have some selective digestion or fermentation of some sugars. So we are using the bacteria of sugar and they are fermenting or oxidizing, then we can say that this particular bacteria might be of this genus. So these are the various kind of enzymatic reaction we are going to perform or we are utilizing for performing various biochemistry or biochemical tests. Now, with this background, we'll come to this four important tests. The first, that is known as KOH test. KOH, that is abbreviation of potassium hydroxide. So KOH test, how we can perform, that we'll see. This is generally considered as alternative to gram staining. If you are not able to perform the gram staining, then by performing KOH test, you can tentatively say that organisms are gram positive or gram negative. For performing this test, you require 3% KOH solution. This is important, you have to remember, along with percentage, because 3% KOH, 10% KOH and 40% KOH. These are the three concentration of KOH generally utilized in microbiology for identification of bacteria. So 3% KOH is used in KOH test, and sometimes it may ask in your exam. So along with pure culture, these are the two things required to perform KOH test. The procedure is very simple. On the clean slide, you require to take a drop of 3% KOH, then take a pure culture with the help of platinum loop and mix this KOH solution with this colony till 1 minute, and intermittently you have to lift the loop. If you are lifting the loop and it will produce the string or thread like viscosity, then you can say that your test is positive. Okay. So why this happens? Because the cell wall composition is different, and cell wall composition is different. That's why that formation of string, that is different. So you have to find out that what is the difference between gram positive and gram negative cell wall, and why some group is giving positive where some group is giving negative. So if you are performing this test, then you have to observe the string formation. So string formation is like this. So once you are mixing your KOH solution with colony of bacteria, and you are lifting after 50 or 60 seconds, then it will form string like this. If string is produced, then you can say that your taste is positive. If string is produced, you can say that your taste is positive, but bacteria is gram negative. If string is not produced, then you can say that taste is negative, but bacteria is gram positive. So this thing you have to remember, because what will happen that when you are performing this test, if taste is positive, then you can say that the bacteria are gram negative. And if taste is negative, means there is no string formation, then you can say that organism is gram positive. So this you have to remember. There should be no confusion in interpretation of your result. So this is in brief about KOR test. Now moving to the next test, that is about Catalase test. So now you have to remember here, ASE, if anything suffix with ASE, that indicate it is a name of enzyme. Okay, so here catalase, it end with ASE, that indicate it is a name of enzyme, and we are targeting that particular enzyme in our test. So now we will see that how this test is performed. Generally in this test, we are using hydrogen peroxide. Again, it is a 3% hydrogen peroxide. I forget to write, so you have to remember. 3% hydrogen peroxide we are utilizing as a substrate. Enzyme is catalase, and the product is oxygen gas. So what will happen, if you are giving 3% H2O2, you are mixing with bacteria. If that contain the catalase enzyme, then catalase enzyme is act on 3% H2O2. That H2O2 will break down it into H2O and O2. So this O2 is a gas, and that will produce the bubbles. So whenever you are observing the result of catalase test, then the ultimate result is a bubble formation. So in KOH, there is a string formation. In catalase, there is a bubble formation. Now you have to be very conscious, because this bubble formation is for short or fraction of time. If you are doing or chatting with your friend and by well mixing, so you may miss the bubble formation and you may give the false negative result. The principle behind this one is catalase enzyme. That catalase enzyme is generally produced by this many bacteria which are performing, or we are utilizing aerobic respiration. So this hydrogen peroxide, that is a toxic byproduct of aerobic respiration, and if that is not removed from that bacteria, that may lead to the alteration of DNA, or sometimes that may cause the death of or mortality or death of the bacteria. So nature had give some mechanism to remove this toxic waste from the bacterial metabolism by catalase enzyme. So you can see this enzyme particularly use is important, because there are earlier I had told that there are very closely related organisms. For example, gram positive cocci. If you are observing gram positive cocci, then it might be a staphylococcus or streptococcus. So in order to differentiate these two, staphylococcus and streptococcus, the catalase is considered as an important test. And if you are performing catalase test, if it is giving positive result, then this is an indicative of staphylococcus. If it is giving negative result, there is no bubble formation. This indicates that your organism is streptococcus. One more difference on morphology of bacteria, that staphylococcus are present as a bunch of grapes. Okay, so જુથ્થાની અંદર દ્રાક્ષના ઝુમખા જેવા દેખાય, whereas in streptococcus they are present in a chain. Okay, so chain might be short or long. So these two characteristic will help to differentiate staphylococcus from streptococcus. Similarly, if you are performing for gram negative bacilli, then the gram positive bacilli like Bacillus and Listeria, they are giving positive result. In contrast to this, the Clostridium group gives the negative result. Now at the end, there is a general statement that all Enterobacteriaceae contain various important genus like Pseudomonas, E. coli, Proteus and others. So this all Enterobacteriaceae, they are considered as catalase positive, and this helps to differentiate other gram negative bacilli. So the utility of catalase test is to distinguish closely related bacteria, and that helps a lot in final identification of bacteria. Now moving to the next, that how you can perform this catalase test. So there are different methods. You can perform this particular test on the slide, even you can perform this test on capillary, even you can directly add the reagent over your agar or the slant where bacterial culture was performed. So you can perform on by any ways, but the ultimate result is the same. If you are performing on slide, then also you can see the bubble. If you are performing on tube or capillary, then also you can see this one. So ultimate result is the same, but you can perform as per your convenience and as per your requirement or availability of reagents. Now how we can perform this one? So there is a short video along with some caption and detection. So you can see that how we can perform the catalase test. So now before starting this one, I would like to tell that you can you can perform catalase test with ready-made disc also, but here it is a demonstration of catalase enzyme they had dissolved and used. Now watch this one. So 2 drops were kept. Now we can take the colony by platinum loop also, and we have to mix this colony with 3% H2O2. So while mixing, bubble will form. So that will indicate catalase positive. This is a negative. You can see if you are mixing and there is no bubble, then you can say that test is negative. So it is very simple test. Bubble is formation, means test is positive. If bubble is not formed, then test is negative. Now moving to the next, that is you need to take some caution or precaution while performing this test. The first is try to avoid performing this test using blood agar. And even you require to perform this test from the blood agar, then try to avoid to take the agar, because blood contains these enzymes by default, and if you are picking up the agar while taking the colony, that will give false positive result. The second is, if there is a old culture, then even though this is a positive, that may give the negative result, and that will lead to the false negative result. So this precaution you have to take in mind while performing catalase test. Now moving to the next, that is oxidase test. So oxidase test, again ASE, that is a suffix that indicate that this is a name of enzyme, and that can perform on to identify the presence of oxidase enzyme in particular test organism or not. So here cytochrome oxidase, generally this enzyme is present in bacteria, and this particular enzyme is identified based on providing some substrate. Once that substrate will break down, that will produce some color change. So the procedure is again simple. You have to take your pure culture over the platinum loop, and you have to rub that pure culture on oxidase disc. Now this oxidase disc might be ready-made, or you can prepare inside your laboratory by adding the reagent. So once you will rub this colony over the oxidase disc, then you have to observe them for color change. So here purple color formation is considered as a positive test, and no change in color is considered as negative. But once again, you have to remember that the color change should be quick. So color change should be within 5 to 10 seconds. Then only you can consider test as positive. If color change after 1 minute or 2 minutes, then you can consider that test as negative. So time is also important, and color change is also important for interpretation of result. If there is no purple color, you can say that test is negative. And again, there is one general statement that anaerobes are oxidase negative, because anaerobes does not require cytochrome oxidase, or oxidase enzyme is not present, and because of absence of oxidase enzyme, generally this particular group of bacteria are oxidase negative. Now again we will see that how this particular test is performed. So here test is performed by producing or by developing oxidase disk inside the laboratory by dissolving the oxidase enzyme by solution, and they had performed the test. But the same test you can perform by ready-made oxidase disk. Okay, principle and procedure will same. Now we will see that how this test is performed. So this is the enzyme vial. Enzyme vial. Enzyme is first of all they are resuspended. A solution is prepared. The sterile disc is already kept in the laminar or in the petri dish. Now after dissolving, you have to add this oxidase on the filter paper or oxidase disc. So this oxidase reagent now that will add on the filter paper, or you can say oxidase disc. Now once it will get absorbed, then you can move to the next step, that is addition or rubbing of your pure colony. Okay. Now this filter paper contains the substrate. Now we are adding the bacteria. So if that bacteria contain oxidase enzyme, that will give a purple color. Okay. So oxidase purple color within 5 to 10 seconds, oxidase positive. Now we are sticking the second bacteria. So no color change, that will indicate that oxidase negative. So Pseudomonas is positive and E. coli is negative. So this is in brief about the procedure of oxidase test. Now moving to the next, that here also you should take some caution or precaution. For example, oxidase test generally should not perform by picking the colony using the platinum loop, using the loop containing iron. So whenever you are performing oxidase test, you must ensure that your loop should not contain iron or it should not made up of iron, because iron is directly giving false positive result. So generally it is advisable to use the glass rod or plastic or platinum loop to perform the oxidase test. Nowadays, even though we are telling that it is a platinum loop, they are made up of nichrome. Okay, so whatever loop we are utilizing in laboratory, they may not be of platinum loop, even though we are calling them as a platinum loop. They might be made up of nichrome, and nichrome contains some sort of iron. So such loop might give false positive reagent. So before performing this, you should ensure whether it is a platinum loop or nichrome loop. The second thing is, when you are performing this test by picking the colony from non-selective media, then your media should not contain glucose and nitrate, because this particular reagents or this particular agent, they are also interfering in oxidase test. Now, knowing the last test of today's class, that is OF test. OF, that is the abbreviation of oxidation and fermentation. So if you will recall the metabolism of bacteria, they are broadly divided into oxidative metabolism and fermentative metabolism. So oxidative metabolism means those metabolism which relies on the presence of oxygen. They require oxygen for performing particular metabolism. In contrast to oxidation, fermentation is a type of metabolism where presence of oxygen is not required. The entire metabolism performs under the absence of oxygen, and that is known as fermentative metabolism. So by utilizing or by performing this test, we can say that this is an oxidative metabolism or fermentative metabolism, or you can say that organisms are classified based on oxygen requirement. So sir might have covered about aerobic, anaerobic, fermentative and others. Okay. So this thing we are targeting by performing the OF test. So how we can perform the OF test? The first thing is what we require. we required is OF medium. So OF medium is ready-made medium. Generally they are semi-solid in consistency and that contain glucose as a taste sugar. And this taste sugar generally utilized by either oxidative metabolism or fermentive metabolism, and that will leads to the production of acid and gas. And this acid and gas generally we are targeting or we are detecting by adding pH indicator, that is bromothymol blue. So bromothymol blue is generally green in neutral pH, that is around 7.1 pH, but it will turns to yellow at around pH 6.1, or you can say at acidic pH. So if glucose is utilized by your target or taste bacteria, then this will produce the acid and this will turn from green to yellow. The procedure is again very simple. Here you require two test tubes that have OF medium. You have to boil this tube so that whatever oxygen is there, that will comes out, and now again you have to cool it down. After cooling, you have to inoculate your test bacteria, and one tube that is marked as a fermentation test, that must be sealed with sterile mineral oil or paraffin or some suitable alternative. It must be sealed so that there is no presence of oxygen and fermentation occurs or not, that we can observe. In contrast to this, the second test tube is marked as an oxidative test, and there is no need to cover with this oil or other material. So oxygen is present and oxidative metabolism will perform. And after inoculation, you have to incubate this tube at appropriate temperature for overnight, and then you can go for observation. So if you are performing OF tests, the observation is like this. The acid production is indicated by color change from green to yellow, because green, that is present as a neutral pH, and yellow at the acidic pH. Acid production and gas production both are possible at the simultaneously. And third, there is a no change. No change means they might be non-reactive. So you can see here green, this is oxidative, this is a fermentative. So both remain green means both tests are negative. Here both, this is oxidative tube, this is fermentative tube. Okay, this black indicate that paraffin is added. So both are yellow means both has metabolism, both turns to yellow, that indicate that both tests are positive. And the last, that in oxidation there is yellow coloration means oxidative test is positive, whereas there is no color change in fermentation, so there is a fermentative negative. So this is general observation you have to perform in OF test, and based on this observation you can interpret this organism in three groups. You may tell that they are the fermentative if the reaction is like this. You may tell that they are oxidative if reaction is like this, and there are some organisms they are considered as a non-reactive because there is no fermentation, no oxidation. This non-reactive might be possible in two cases. One, if organisms are fastidious, they cannot perform or they cannot grow in this basal medium, that is OF medium, then in such cases you have to go for addition of some supplements like serum and other thing. And second thing, there are some organisms, they are by default, they are non-reactive to this particular glucose. So they cannot utilize glucose, and that's why they are not giving reaction in this test. So you may require to go for some other sugar. So this is the example that Pseudomonas are generally giving oxidative type of result, whereas Aromonas is giving the fermentative type of reaction, and Bordetella, particular Bordetella species, they are considered or they are giving non-reactive reaction. So this is in brief about the observation, procedure and result of OF test. Now this is the last slide. I would like to thanks, and before thanks there is one more assignment for you, that you have to, okay, I had covered up this principal, procedure and interpretation, so no need to go for this thing, but you should know that KOH means string formation, catalase means bubble formation, oxidase means blue color formation, OF means color change. So you should know the result and their interpretation. But the most important assignment you have to complete that you have to analyze the result of this test for veterinary important pathogenic bacteria. Means you have to prepare one table that if Q was positive, then this organism, if Q was negative, then this organism. Catalase positive, this organism, negative, this organism. For oxidase also like that you have to prepare, and for AF there is a bigger table will form because there are number of combination along with other tests. So that table you can prepare and you can say that by performing these 4 tests, what type of bacteria or what genus of bacteria you can identify. So with this, I will end up today's class. Once again, I thank you everyone for listening me and giving me this opportunity to present introduction to biochemical test. If you have any query, you can divert it on this mobile number and this email ID. I am happy to answer it. Thank you to one and all. Thanks.